Hi grade 12 learners, welcome to today's Life Sciences lesson. My name is Radine Kruger and this lesson is brought to you by the Gauteng Department of Education in collaboration with Bono Discovery Centre and broadcast by Bakama Research and Development. We have a WhatsApp line where you can send questions and maybe pictures, but you can't send voice notes or videos. And the number is 063-629-3655. Okay, so today what we're going to do is we're going to have a look at homeostasis in humans, but particularly we're going to have a look at the content that you covered in grade 11. So we're going to do a little bit of revision of what you learned last year. And remember, the reason why we are covering that is because you will be tested on it in this year's exams. Okay, so we're going to have a look at the concept homeostasis, and if we have a look at the word, we can see, or we can try and figure out what the term means. And stasis, remember, means to keep something balanced or equal, or to keep it the same. And then homeo is the environment. So basically what your body does is it's doing kind of a balancing act to try and keep whatever nutrients, substances, whatever in your body at a balance. So we don't want too much or too little of something, and your body needs to keep that in balance. And the information that it receives to tell you that something is out of balance comes from the environment. And it can either be from the external environment, from outside, for example, or it could be from your internal environment, which is inside your body, from your blood, or from hormones, or mostly from your blood and your tissue fluid. There's things that are, the levels change. Okay, so let's have a look at the term, what it actually is. And if we have a look here, we see it's the process of maintaining a constant internal environment. So in other words, all this nutrients and substances and waste products in your blood and in the tissue fluid, that is known as your internal environment, must be kept in balance. So that then enables the body to function efficiently. Okay, so even if there are changes that happen in the external internal environment, your body will still function as it should. Right, so and that is the process of homeostasis. As I said, it's a continual balancing act. All right, now what is important is that the internal environment, so that we understand what the internal environment is, and that refers to the blood and the tissue fluid. So in other words, it's all the fluid in the blood. And then remember, every single cell doesn't have its own blood vessel because our bodies consist of so many billions of cells that we then have capillary networks in the tissues, and out of that squeezes tissue fluid that contains substances required and that also then removes substances that we don't require. Right, so now that is how we or that's the substance that is going to then be where the homeostatic control or mechanism must work. And the way in which it works is by a negative feedback mechanism. Now, a negative feedback mechanism basically is where there's constant feedback between a receptor that picks up the stimulus, a controlling structure, and the effector or the organ or the structure that's going to bring about the change or bring about rebalancing, whatever it is. So that we call a negative feedback system, and that is continually taking place in our bodies. Now, that can either occur via your nervous system. When we learned about the nervous system, we learned that it is a coordinating system, and it can also occur via your endocrine system. So both of them then function in our homeostasis or homeostatic control in using a negative feedback mechanism. All right. So what is important to know is that a negative feedback system, negative doesn't mean something bad for your body. Doesn't mean that it's a negative thing and so it's harmful to your body. It's not. All it means is that there's continual feedback between the receptor, the controlling structure, the effector. So there's, as soon as a substance or something goes low, it's out of balance, then the stimulus gets to the controlling center. The controlling center will then stimulate a response, and it will bring it back to normal levels. And then if it goes beyond the normal levels and goes too high, then the receptor is going to pick that up again, and then we'll ensure that we don't have either too low or too high, but we rather keep it in a balance. Okay, now what is important about a negative feedback system? Very simply, it consists of three main components. And it's important to remember that the three main components are a receptor, as I said, and we learned earlier on when we did our sense organs that a receptor is some structure in your body, sensory structure, cells in your body that then pick up stimuli from the environment. So it can be something like from the outside where the temperature changes from hot to cold, or it can be from inside your body where you've got too much carbon dioxide in your blood, or you've got too much oxygen. So there's a receptor that is then able to pick up the changes. And those changes, it doesn't help to just pick up the information that there is a change or there's an imbalance. It must go to a controlling center. For example, your brain. Okay, and your brain will then analyze that information and say, okay, there's too much or there's too little, and then it will send a message to the organ or structure that's going to bring about a change in the balance, and hopefully bring whatever it is back to normal. So you've got the three main components, which is a receptor, your control center, and an effector. So if we have a look at this diagram here, which is an example of a negative feedback loop or mechanism, you can see the body there in the middle is at a good temperature. Then if it gets cold, the body temperature decreases. So what happens is your body then starts to shiver, or you shiver, and your body starts to warm the body up so that it can increase the temperature. Now, if the temperature goes too high when you get hot, then it initiates a cooling process, and the cooling process is then going to cool your body down and try and get your temperature back to normal. Okay, so that's very basically, or very simply, a negative feedback mechanism. All right, now in Mind the Gap, we've got 7 steps that we usually teach so that you remember the general or the generic sequence of a negative feedback mechanism. And we call them steps because then, they follow each other. They have a sequence. So if we have a look at the first step in a negative feedback mechanism is your body is going to detect an imbalance. Okay, so it's going to pick up, your receptor is going to pick up that something's not in balance. So an imbalance is detected. Secondly, or the second step, the control center is stimulated. And when the control center is stimulated, it's going to respond. And then it then sends a message to the target organ or organs so that the target organ then responds. And when the target organ responds, it's going to oppose or reverse the imbalance. And in that way, the balance is then restored. So if you know those 7 steps, if you learn those 7 steps and you know them, it doesn't matter which example we give you in the exams or you are given in the exams, you would then be able to adapt that scenario to these 7 steps, and then you would be able to answer. So it's very important that you do learn this, and it is in Mind the Gap where you can learn the 7 steps, the generic steps. Right. Okay, so what's important is a negative feedback mechanism basically controls the following in the body. So the ones that we learned last year in grade 11 would be your blood glucose levels. So it's very important that there's a balance maintained. carbon dioxide levels in the body, and then also osmoregulation, which is your water and your salt levels. Okay, so those were the topics that we covered in grade 11, and you need to revise them because you will be tested on them for grade 12. Okay, so now if we apply, so we can take any of those three examples and we can apply it to our general sequence of events in a negative feedback mechanism. So for step 1, I could say, let's have a look at blood glucose levels. I could say there's too much blood glucose, so there's receptors that will pick that up. That message is then sent to your control center, and the control center then stimulates or respond, and it stimulates the pancreas to release insulin, and then the blood glucose then will drop, so the balance is restored. Okay, so that's why if you know those 7 steps, it doesn't matter which example you're given, you would be able to cover it. Okay, so let's just have a look at the terminology quickly. And remember, terminology is very important because if you don't understand the language of life sciences, you're not going to be able to understand what a question is asking you, and then you won't be able to answer very well. So the first term we're going to have a look at is what is an endocrine gland, or what are endocrine glands? And endo means inside. So those basically are glands that are ductless. So in other words, they don't have a little tube or a little duct that will carry the substance they make outside of the gland to wherever it must go. So they then, inside the gland, secrete that substance straight into the bloodstream, which is in the blood vessels inside the gland. So they then secrete the hormones directly into the bloodstream, whereas exocrine glands, and remember exo means outside or external. So those are exocrine, which means that they have a little duct. Okay, so they would have a tube or a duct that carries the substance. For example, your salivary glands or whatever is a little. Your salivary glands are under your tongue, and then they carry the saliva via little ducts into your mouth. Okay, so that is an exocrine gland. So it's important that you know the difference between those two. And then the other important concept is hormones. And hormones, they are organic chemical messengers. So they are chemicals that the endocrine glands produce, and they are secreted into the bloodstream by the endocrine gland. And remember, they're not going to work at the endocrine gland. They're going to go and work somewhere else in the body. And we call that place where they go and work, we call that the target organ or structure. Okay, so those are the basic concepts or the basic terms. And here you can see it's just a comparison between exocrine and endocrine. So your endocrine glands, they are ductless and they secrete directly into the bloodstream. So there you can see inside of, in this diagram, the cells there that produce the hormone are going to secrete it straight into the bloodstream, which would surround the little cells. Whereas an exocrine gland, they have little ducts. So here you can see this is actually a sweat gland, or it can be any gland in the skin. And what it does is it produces the substance inside the gland, but then there's a little duct or a little channel or canal that the substance then is going to move to wherever it needs to. And this is on the outside of the skin, on the skin surface. Okay, so that is the difference. And then remember that exocrine glands do not secrete hormones, so that's important. So we don't ever find hormones coming from an exocrine gland. They all come from endocrine glands. Right, okay, then important glands and hormones in the body, and the ones we're going to look at today is we're going to have a look at specifically the hypothalamus, which secretes ADH, and we're going to have a look at the pancreas, which then secretes those two hormones, insulin and glucagon. And we're also going to have a look at the adrenal glands and see they secrete the two substances, adrenaline and aldosterone. But we're going to have a look at aldosterone. Okay, so those are the main glands or structures that that are involved in the negative feedback mechanisms that we're going to cover today. Right. Okay, so let's have a look specifically at homeostatic control of blood glucose. Okay, so we know that there's a negative feedback mechanism that works between the pancreas, which is that structure there, and the liver. Okay, and it's going to then regulate the amount of glucose that we find in our blood. Right, so we'll find that after a meal, so once we've eaten something, okay, once it's digested and that those nutrients have been absorbed, then our blood glucose levels are going to increase. And you'll know that after break when you've eaten and then you go to class for the next lesson, you feel like your eyes are so heavy, you want to sleep. And that's because what happened is at lunchtime, and then you've eaten something, carbohydrates, and your blood glucose levels have increased. And so what did your body do? It detected this change and it released insulin. And the insulin is now removing that excess glucose. And when there isn't glucose in the bloodstream, so if the level goes too low, that's when we don't have enough energy and then we're very tired. Okay, so, and that then is how a negative feedback system works. So then after a meal, our blood glucose levels will increase because there's more glucose that has been absorbed from the small intestine into the blood. And the pancreas then detects an increase in blood glucose and it releases the hormone insulin. Okay, so it's insulin that is going to be released, the hormone that is going to be released when your blood glucose levels are too high. And what that does is it stimulates the liver cells and your muscle cells to either absorb glucose or to use the excess glucose. So it removes the glucose from the bloodstream and it's absorbed into the cells, and then it's converted then into what we call glycogen. And glycogen is a stored starch in animal cells. So it's converted into glycogen, and so then our blood glucose levels will drop. So glycogen is stored in the liver and your skeletal muscles in the body, and the body cells are also stimulated to use a little bit more glucose. So then that then will return your blood glucose levels to normal. Okay, so now when you've been busy, active, what happens is now your blood glucose, so it's a while since you ate, and the insulin has kicked in and it's removed all the excess glucose. Now your blood glucose levels go a little bit low, so they decrease. And as soon as that happens, then the pancreas again is stimulated, and now it releases a second hormone called glucagon. And what I want you to do is to know the difference between the hormone glucagon and the substance glycogen. Because if you misspell them and you combine and you say glycagon, for example, in the exam, we're going to mark it wrong. So you must know the difference between these two terms. Glucagon is the hormone that is going to be stimulated or released, and it's going to stimulate the decrease or an increase in the blood glucose level. So in other words, it's going to stimulate the breakdown of glycogen, which is our stored starch or stored carbohydrate in our cells, cause it to break down and into glucose molecules again, which once they are small enough as glucose molecules, they can then diffuse back into the bloodstream, and that will then raise your blood glucose levels. So it's very important that you know and you don't confuse the word glucagon and glycogen. Okay, and then once your glycogen has been broken down and the glucose goes back into your bloodstream, that will then increase the blood glucose levels back to normal. All right. So if we have a look at the diagram, and it was the same as the diagram before, so you can see there's a negative feedback between the pancreas and the liver, and what happens is if the blood glucose levels get too high, the pancreas is going to secrete insulin, and insulin is then going to stimulate the muscle cells to take glucose out of the bloodstream and convert it into glycogen. Now what happens is it lowers the blood glucose. But that can then lead to low blood glucose levels. So your glucose levels now go very low. So that message now goes back to the pancreas. Okay, and this is where we say there's a constant flow of information between the different organs. So that's where your negative feedback mechanism comes in. So now the pancreas has detected that the blood glucose levels are low. So that's now going to stop secreting insulin. We don't need any more insulin because already our blood glucose levels are low, and it's now going to secrete glucagon. And when the glucagon is secreted, that then goes via the bloodstream to the liver cells, and it's going to stimulate the liver cells to now break down glycogen back into glucose, and that then goes back into the bloodstream and it raises our blood glucose level. So we've got this constant balancing act happening in the body, okay, to maintain our blood glucose levels. All right, so and have a look at the different diagrams here. Here we've got kind of what we call a flow chart. Here we've got a diagram, because you don't know in the exam what example or how the examiners are going to ask you. They might ask you to even draw a flow chart like this, or they will give you a diagram and possibly leave out the labels and then ask you to fill in the information. So it's important that you look at different versions of our negative feedback mechanism in the glucose blood levels, but basically they're the same. So here in the middle you can see there's normal glucose levels. Then what happens is I eat something and my glucose levels are going to increase. And so what happens is the response there is my pancreas secretes insulin, and insulin is going to go to the liver cells and the muscle cells, and it's going to cause the conversion of glucose into glycogen. And then that's going to then cause my blood glucose levels to decrease. And they then hopefully go back to normal. But now if I don't eat for a while, it's a few hours before I eat again, now my blood glucose levels go too low. So now again that information is received. So it picks up, no, the other way around, my apologies, not that way. So it's here, it picks up that the glucose levels have now decreased. And that then that message goes to the pancreas. Pancreas is now going to secrete glucagon. And glucagon is going to stimulate your liver now to break down that glycogen so that it goes back into glucose molecules. And when it's glucose molecules, it can go back into the bloodstream, and that's going to increase your glucose levels back to normal. All right. Okay, so now people who are unable to maintain, or their bodies are unable to maintain their blood glucose levels would suffer from a disease called diabetes mellitus. And basically what it is, it means is that there's sweet urine. All right, so it's a disorder that is caused by high blood glucose levels. So what happens is there's a lot of glucose in your blood. And when your kidneys are now functioning, when the nephron is functioning, usually in somebody that doesn't have diabetes, their bodies would reabsorb all the glucose that is in the filtrate that goes into the kidneys. Now what happens is when there's too much or there's very high levels of glucose, the kidneys don't reabsorb all of the glucose. So that glucose, and remember glucose is a useful substance, so your body doesn't want to excrete it and get rid of it, waste it actually. So what it does is it wants to reabsorb all that is in, all the useful substances from the filtrate back into the body. So what happens here is that the glucose is too much glucose, so some of it remains in the urine, and then that's how doctors are then able to detect. It's one of the ways that they can actually test the urine to see what the glucose levels are. So somebody with diabetes, the symptoms usually are they have excessive thirst, so they're always thirsty. They also have high blood sugar levels or blood glucose levels. They might lose weight. They're always tired because their bodies are not using the glucose that they are taking in effectively. Wounds heal slowly, so the body just isn't working correctly. They might have blurred vision. They need to go to the bathroom very frequently because they're thirsty, and so they're drinking a lot of liquid, and so continual flow. So there are ways in which we can treat and we can prevent. Usually, a preventative mechanism is to change the diet and the lifestyle. So in other words, you might cut down, the doctors will recommend that you cut down on the amount of sugar and carbohydrates that you take in. And also, if it's type 1 diabetes, then there would be medication that you use that would help you regulate blood glucose. All right. So it's diabetes mellitus is a disorder caused by high blood glucose levels. It does result in increased fatigue or tiredness, dehydration, and lack of energy. Now, there's two types of diabetes mellitus. The first one we call type 1 diabetes, and the other one is called type 2 diabetes. Now, type 1 usually has an early onset. So, in other words, people who have type 1 diabetes, usually the onset is when they are still young. Whereas type 2 diabetes usually has a later onset. So, in other words, as people get older, so they then might develop type 2 diabetes. Now, the cause of type 1 diabetes is usually an inherited disorder, or otherwise the body has breaking down the insulin-producing cells in the pancreas. So it could be some kind of an autoimmune disorder. Okay, and what happens there is individuals with type 1 diabetes, the treatment would be that they would need daily injections of insulin and especially adapted diets. As I said, you would cut down on the amount of carbohydrates and sugars that you take into your diet, but they would need to then inject themselves with insulin daily. Okay, then type 2, as I said, it's usually later onset. So when you sort of get older, what happens is your pancreas, and especially if you've been somebody who, snacked a lot on high carbohydrate and those kind of foods, your pancreas actually becomes tired, your body becomes insulin resistant. It doesn't necessarily produce enough insulin, plus also the cells are unable to use the insulin like they should. So it does not produce or react to insulin, and that's usually as a result of poor lifestyle choices. So those KFC and McDonald's and those kind of things, it's not a very good diet, a very healthy diet. It can lead to type 2 diabetes. Now, the treatment of that usually is maintaining a balanced diet. So there the doctors would encourage individuals to cut out on all those processed carbohydrates, maybe start a little bit of exercise, regular exercise, and then sometimes medication as well. All right, so those are the two different types of diabetes. And here's just an example or a diagram that shows. So you can see in a healthy person, as soon as the glucose levels increase from the stomach, it goes into the bloodstream. If you have high levels of blood glucose, the pancreas is then going to secrete insulin, which goes into the bloodstream, and it causes the liver and the muscle cells to remove the glucose from the blood and convert it either to glycogen or to use it. So that's in a healthy person. Now, in somebody who has got type 1 diabetes, you'll see the glucose enters the bloodstream after a meal, but you can see how much less insulin. And sometimes there's no insulin. Body doesn't produce any insulin, and so it's not able to effectively stimulate the liver and the muscle cells to use all the glucose that is in the blood. And so what happens is then we have an increased level of glucose that stays in the blood. Right, if we have a look at the next picture or diagram, it shows type 2 diabetes. So here you can see again the healthy person, it's exactly the same as the previous one. When there's increased blood glucose levels, the body secretes enough insulin, and the insulin then encourages the cells then to take up glucose and convert it or use it, so that we keep the levels of glucose in the blood at a normal level. However, somebody with type 2 diabetes, again here you can see the glucose levels. Here you see that there is enough insulin, but for some reason the cells in the body become resistant to insulin. So in other words, where insulin would work as a little key in the cells to get the reaction, now the body has become resistant and the insulin isn't working like it should. So there you can see it says the organs are unresponsive to insulin, and we call that insulin resistance. So there's less glucose that's being stored or used, which means that our blood glucose levels stay high. Okay, so that's the difference between type 1 and type 2 diabetes. Right, and then the causes of type 2 diabetes can be high blood pressure. It's very often is linked to high blood pressure, some kind of disease of the pancreas, obesity. Okay, and obesity is a big problem, and it's very often people who suffer from obesity would have type 2 diabetes. Age, you can see an increase in age. So as you get older, there can also be a genetic predisposition. Stress, lifestyle, sedentary means you're not very active. You sit on the couch. There you can see the couch. Some people call it couch potatoes. Unhealthy food. There you can see highly processed carbohydrates. And sometimes medicines. Okay, they can cause it. All right, so now what we're going to do is we're going to have a look at some of the questions that we find in exams, the way in which the examiners ask the content of this. And remember, as we said at the beginning, you were taught this content in grade 11. Your teacher's not going to reteach it. There isn't time, but they will revise it, and that's why we are also doing it now, but you will be examined on it. So it's very important that you do understand the concepts. Okay, so the first question is they give you some information. They say type 1 diabetes mellitus is caused by pancreatic cells which cannot produce insulin. In type 2 diabetes mellitus, the pancreas does secrete insulin, but the body cells are resistant to the effect of insulin, so it doesn't work. To diagnose if a person has type 1 or type 2 diabetes mellitus, the following tests may be conducted. Okay, it's supposed to be following. The blood glucose level is tested after fasting. So in other words, the person is not allowed to eat or drink for 6 hours before the test. So they had to abstain from eating or drinking anything, so that the blood glucose that is in the blood is used, so that then they can see. So if the blood glucose level is above 126 milligrams per deciliter, then they say, or they diagnose that the person is diabetic. Person is then given a glucose solution to drink, and the blood glucose is tested again. The blood glucose level is tested again after 2 hours. The blood is also tested for the presence of GAD antibodies, and the presence of GAD antibodies would indicate that the body is destroying the insulin-producing pancreatic cell. Remember, that's important to distinguish between whether it's type 1 or type 2 diabetes. And then also, another test that they conduct is the levels of C-peptide are measured. So this is a substance that is produced by the same pancreatic cells that produce insulin. So if the levels are below, and here they say below 0.3 nanograms per milliliter in the blood, the person is diabetic. So in other words, if it's very low, then that means there's no pancreatic cells, or the pancreatic cells are not producing insulin. All right, so let's look at the questions. Okay, so the first question they ask based on that information is to name the cells in the pancreas that secrete insulin. And those cells are known as the pockets, or the groups of cells are known as the islets of Langerhans. And that's as much as you need to know. And the second question, explain which type 1 or 2 of diabetes mellitus would be indicated by the following. First of all, the presence of GAD antibodies in the blood. And if we go back to the information, and it said when they test for that, because the presence of that would indicate the body is destroying those insulin-producing pancreatic cells. Okay, so what type of diabetes would that be causing? And that would be type 1. Okay, and that is, is because it would be type 1 where no insulin would be produced because the body is destroying the cells that are supposed to produce insulin. And that then indicates that the pancreas cells are being destroyed or broken down. And then what type of diabetes would be indicated by the C-peptide levels that are below 0.3 nanograms per milliliter? And then it would also be type 1, and that is because a lower than normal C-peptide level indicates that the insulin-producing cells of the pancreas were destroyed. So they're not there. So they can't produce C-peptide, or there's not enough of them to get to the correct amount of levels. Right, then the third question, they ask you to explain, and remember, and we've spoken about this before, where we say an explain question is usually, and it's good to look at the mark allocation because there you need to explain why something is as it is. It's not just basically a description. Okay, so you normally need to give a reason. So here they ask you to explain the insulin levels in the blood of a type 2 diabetic 2 hours after drinking the glucose solution. So, sorry, I left the last line off there. But here, the insulin levels then, remember, they will remain high. So there would be insulin levels in the blood, but because it's type 2 diabetes, the blood glucose levels will also remain high. So remember, the body's not using the insulin as it should, and that is because it's resistant to the insulin. So the pancreas, because the blood glucose level stays high, it will continue to secrete insulin. So what you'll find is that high levels of blood glucose, that message keeps going back to the pancreas. And so the pancreas says, oh, the blood glucose is still too high, and it secretes insulin. So you'd find in a type 2 diabetic, they would then have high levels of insulin. Okay, then question 4, they ask us to give one reason. And when they ask for only one reason, they're usually going to mark your first answer. So make sure that that first answer is correct. Because if you give a second answer and that's the correct one and not the first one, you will lose the marks. Okay, so one reason why a person with untreated diabetes mellitus is expected to be tired all the time. And that is because the glucose that is in the bloodstream is not absorbed and it's not used by the cells. So if I don't have glucose in my cells, cellular respiration can't take place and my body can't make energy for me to actually function. So then that individual is constantly tired. Okay. And therefore cannot be used in cellular respiration or can't be used to release the energy that's in the glucose. And so then the individual, constantly remains tired. Okay, let's go to the next set of questions. Now again here, so they haven't given you a direct or just a level A or a level B type questions. Usually for these concepts, they give us more challenging questions. They usually base it on a case study. So there's information that you need to learn. And so it's always important when they give us a graph like this to have a look at the graph carefully to see exactly what information they've given us, and then to have a look at the stem of the question and see what other information they've given us, and then to answer the questions. So if you have a look here, they say the graph below indicates the effect of glucose concentration on insulin production before and after a meal, and that was measured for a healthy person. So this person's body reacts normally to increased levels or decreased levels of blood glucose. Okay, so if we have a look, here the heading says the effect of glucose consumption on the concentration of insulin over time. And we can see there's 2 line graphs on this graph. There is a key. We can see there's a solid line and then there's a dotted line. So we're going to have a look at the key and we can see what does the solid line indicate and what does the dotted line indicate. And we see the solid line is glucose levels and then the dotted line is insulin levels. Okay, and then on my y-axis, basically it's the concentration of those two substances, whether it be glucose or insulin, that's in your blood. And then on the x-axis, we've got time. Okay. So, and there we can see our blood glucose is at a certain level, and then it increases, and then it starts to decrease again. We can see our insulin levels also stayed fairly stable, and then a little while after the glucose levels started to increase, we see our insulin levels start increasing. So your body got the stimulus that your blood glucose levels are increasing, and that then caused the reaction of the pancreas to increase its insulin secretion. So that would also increase. All right. So the first question they ask us is, during which period of time was the person's blood glucose level constant? Okay, so now we have to look at our x-axis. We have a look at the glucose levels, and we can see that between 7 o'clock and we can use our ruler to show us. Okay, I don't have a ruler, but between 7 and 8, the blood glucose remained constant. Okay, it didn't increase and it didn't decrease. So that would be our answer. And then it says the second question is, what is the glucose level in the blood at 9 o'clock? So then I go back to my x-axis and I would use my ruler, draw a line up, and I can see at that point, and then I take my pencil and my ruler again and I draw a line across to my y-axis so that I can figure out, so where those two lines intersect on the dot, then it will tell me what the amount of blood glucose is at 9 o'clock. Okay, so if I have a look there and I can read it off, it says it's per milligrams per 100 moles, and in this case it looks like it is at 93. Right, then the third question they ask us to describe the homeostatic control of blood glucose levels after eating breakfast at 8. So we can assume that at 8 o'clock, because the blood glucose levels increased, that the person had breakfast then, and that is why the blood glucose levels increased. So what happened in the body once I had had my breakfast? Okay, so what happens is, the first thing, my blood glucose levels increase. Which then means that the pancreas is stimulated to secrete insulin into the blood. So now I've got insulin in the blood, which then goes to the liver and the muscle cells, and that stimulates the absorption of glucose out of the blood. Okay, so that's the glucose is absorbed, and it stimulates the conversion in those cells to change the glucose into glycogen. And glycogen, remember, is a stored starch, so it's not able to diffuse. Okay, so it stays in the cells, and then they are stored in the liver or in the muscle cells. And I'm sure you've heard people talking about their carbohydrate stores, especially those that are athletes. It's good to build up those carbohydrate stores so that when you do a race or whatever, you then have excess glucose that your body can call on and use for the energy that is needed. Okay, so once the glucose is removed from the blood glucose levels decreased, and they went back, they were on their way back to normal. All right. Okay, the next question says, describe the expected changes in insulin level if this person was diabetic and untreated. Okay, so let's see what would happen then. Okay, so what happens then in a person who is diabetic, the insulin levels would remain low. So they do not rise after the meal, or they don't increase. They would stay around about the same, and it's quite low. All right, so go back there. So that's basically what happens. So that because the person is diabetic, the insulin levels remain low, and because they say they are untreated, so they're not taking medication, they're not taking insulin, they are unmedicated, so then that would remain low. Right, let's have a look at the next question quickly. Here, in this case, again, they've given us a graph, and we have a look at the graph. We can see on the x-axis, it's the level in the blood, And then on the y-axis is level in the blood, and on the x-axis they've got morning, noon, and evening. They don't have specific times, but we can see that there's two line graphs, and they say the black line is blood sugar and the red line is blood insulin. And again, there they give us some information. There they say breakfast was taken, so we can see the blood sugar increased, and then so did the insulin, which then caused a decrease. Then we had lunch, and the same thing happened, and then dinner, the same thing happened. All right, so let's have a look at the questions. So first of all, what happens to the blood sugar level every time the person eats a meal? And we saw that each time after a meal that it increases. Then why does the insulin level only increase after the glucose level increases? Because remember, that is the message. So when the body picks up that there's increased blood glucose levels, and that would only happen after it's been digested and absorbed, then that would then stimulate the pancreas to secrete insulin. All right, what does the straight line represent? So we can see there's a straight line that goes through the graph here, and that basically indicates normal blood glucose levels. So you can see this person's blood glucose fluctuates quite a lot, which is not always ideal because it doesn't keep your energy levels stable. Right, then the fourth question is, describe the effect that insulin has on the glucose levels in the blood. Let's see. And that is, as soon as there is insulin and your glucose levels increase, it stimulates the conversion of the excess glucose into glycogen, and that then would basically lower the amount of blood glucose and therefore return the levels to normal. All right, and then the last question, I think this is the last one, is there's an extract that they've given, diabetes and treatment and management. There's two forms of diabetes are found in humans, namely type 1 and type 2. Type 1 diabetes, the body's immune system mistakenly sees the insulin-producing cells in the pancreas as foreign and destroys them. And remember I said to you it's kind of like an autoimmune, so your body is attacking itself. And then people with type 2 diabetes are able to produce some of their own insulin, but often it's not enough, and that's overeating, and especially highly processed, rich in sugar, causes repeated stimulation of the pancreas, which responds by secreting large amounts of insulin. The excess insulin decreases the target cell's ability to respond to insulin. Okay, so then treatment is focused on diet and exercise. Okay, the first question they ask us, name one body fluid that can be used to test for the presence of excess glucose in the body. Remember we said there would be high glucose levels in the urine, so that would be one of the substances, but they can also test your blood. Okay, so they can either use blood or they would use urine. And then they say give two target cells in the human body that will be affected by an excess of insulin, and that would be your hepatic or your liver cells, and the other one is your muscle cells. Right, and then explain the consequence for type 1 diabetics when their immune systems destroy the insulin-producing cells. So for them, the body cannot reduce blood glucose levels, and that causes the blood glucose remains high. So what they need to do is they need to inject themselves daily with insulin, and that would then return it to normal. Okay, I think that's all the questions that I have time for now. Griselda van Wyk is going to take over and do the next two concepts. But remember, the more exam questions you practice, the better you will do in the exam. And good luck. Thank you.
Hi, I'm Griselda van Wyk, and I just want to thank Radine for that very interesting presentation of insulin and glucagon and glycogen. Now we're going to go and look at your breathing mechanism, right? So we're going to look at the homeostatic control of carbon dioxide levels. Now let's just think, why do you have carbon dioxide levels? Well, just think about it. You're breathing and you are breathing carbon dioxide out. Why? Because of respiration taking place in your body. Your cells need energy. Where do they get the energy? When you are stuffing all that carbohydrates in your face, you're eating breakfast, afternoon, evenings, chips, burgers, whatever, that is fuel for your body, right? So what your cells do is it takes that food, glucose, it breaks it down into energy, and CO2 or carbon dioxide is one of the byproducts. The other one is obviously water. So when you breathe out, you will feel like moist on your hand, right? That's the water that's coming out from cell respiration. So after all this cell respiration that's going on in your body, there's a lot of CO2 building up in your blood. And it can't just keep on building up, because you will go into a coma and die. And that's where homeostasis comes in. It regulates the amount of CO2 and it makes you breathe out the CO2. That's very important, right? So, let's have a look how it works. Firstly, I'm just going to show you this here. When the carbon dioxide levels in your blood increases, now it's important when you answer a question in the exams, talk about carbon dioxide levels in the blood. Right, you can't just say in the body or just carbon dioxide levels increase. No, it's in the blood. That's very important. If you do not say the in the blood part, they don't give you the mark. Right. So when you now obviously just living, you're breathing, you're doing your thing, the carbon dioxide levels increases in your blood. Right. And what happens is that it forms an acid in your blood, carbonic acid, right? And that decreases the pH of your blood. And when that happens, normal cellular activities and stuff in your body can't take place. So it is then detected. That's like a stimulus. It is detected by the receptor cells in your carotid arteries, which are arteries here in your neck, as well as in your heart. You've got the aorta arc, right? There's receptors in there that's picking up that there's something wrong with the pH of the blood, right? So then it will send an impulse to the medulla oblongata, right, of the brain. So now this kind of looks familiar because we've got a stimulus, we've got a receptor, and we've got an impulse being sent to the control center, right? Just remember that. So now what does this medulla oblongata do? It's not just sitting there minding its own business. No, it immediately sends impulses to the effector organs, which is now your intercostal muscles, which is in between your ribs, and your diaphragm. That's your breathing muscles. So that you start to breathe faster and deeper, because you need to get more oxygen in and more CO2 out of your body. You need to get rid of all that CO2, right? And also your heart muscle will start to contract faster, right? So you will be pumping blood faster through your veins. Why? You need to get the blood to the lungs so that you can get rid of all that CO2, right? So the CO2 will then be removed faster and more CO2 will be removed, right? Let me just get my pen right to go on. So that's the part that I just said now. And as more carbon dioxide is leaving your body, it's being exhaled through breathing, your carbon dioxide level in your blood will then return to normal, right? And that's basically it. It's not a very complicated situation. You just need to know the steps of what will happen in this situation. Now, in grade 12, you are lucky enough to have the Mind the Gap booklet, like Radine also referred to that. And Mind the Gap has the most wonderful, wonderful summaries of the negative feedback mechanisms and homeostatic control. This table is taken from Mind the Gap, right? And if you study this, if you learn this off by heart, you study it, you know it, you can answer any CO2 question in a question paper. So let's just have a look again. So step 1 will be if the CO2 levels increases in your blood, right, above normal. And emphasis in the blood. Okay. Firstly, your receptor cells in your carotid, there's the carotid. You can see I've given you a picture there. There's the other one there. There's receptor cells there. There's the aorta arc over there. Now, the receptor cells in there are stimulated, right? So, they send a impulse to the medulla oblongata. Now, for the matric learners, this looks very familiar. I hope so, because in June you wrote exam about the brain and the nervous system. I really do hope you recognize this picture. Right, now the medulla oblongata is over there, and that will then send impulses to your breathing muscles, like I said, your diaphragm and your intercostal muscles, and they will start to contract more actively and increase the rate. Rate refers to how faster, right? Faster. And depth of breathing, more the volume will be increasing. More air will come into your lungs, right? The heart will also beat faster. The blood will be pumped faster through your body and faster to the lungs so that the CO2 can get out faster. More CO2 is taken and exhaled from the lungs, which I just said. And then step 7, your CO2 level in your blood will return to normal. Right, so, and people, this is almost like a memo. If we mark, I've been marking for many years now. This is almost the memo we use when we mark, right? It also depends on the wording of the question. That's also sometimes make it a bit different where we concentrate on, but usually the memo is, yeah. Right, so that's why I say mind the gap with your endocrine system. People, very important, use it. Nice resource. Right, let's look at a question from this section. A learner is training for a race by running 20 kilometers every day. People, I always dream about this. While I'm lying on my couch, like she said, a couch potato with my bag of chips, carbo loading, doing a binge watching of Netflix movies. I also sometimes dream of doing this 20 kilometers. I was like, I can almost visualize myself before I go off to sleep. But there are actually people who does this, right? They are running for fun. Anyhow, now this question wants to describe how carbon dioxide produced in the body during training is controlled to maintain normal levels. Now we just spoke about it, right? So firstly, because the exercise, the carbon dioxide levels in the blood will increase. Why? Because now there's more cellular respiration going on in your muscles. You need energy, right? So the respiration rate is going up. Need energy. Now, if the respiration rate is going up, more CO2 will be secreted into your blood stream, and that's why it will increase in the blood. And then I see they missed a step here, because you have to talk about the receptor cells in the carotid arteries and in the aorta, right? And that will send an impulse to the medulla oblongata, which is stimulated, and that will then not send a message. Please don't talk about a message, rather talk about an impulse to the heart and the breathing muscles. The heart will obviously now beat faster, right? And why? Because now the blood with the carbon dioxide will go to the lungs faster. So the heart is pumping, getting that CO2 to the lungs quicker. Because you're running, you can't get an overload of CO2, you'll pass out. And then people overtake you and win you. You don't want that. Right. Also, your breathing muscles will contract faster. Like I said, the rate and the depth will be increased. So more oxygen will then be removed every time you breathe out. And then your carbon dioxide levels will be decreased back to normal. Right, and that's how it's done. Easy peasy. Next question. An investigation was carried out on a person in which the volume of air taken in at each breath and the number of breaths per minute were measured at rest and after running. Yeah, this is now athletes, the guys who actually run, they would know about how you're taking your resting heart pulse and, no. all those things. Very impressive with the watches and everything, you know. Anyhow, but at this experiment, what they did was at rest, just before you are running. Like I'm always at rest on the couch, but it's just before you're running at rest, you are inhaling 450 cubic centimeters of air, right? And that's about 450 milliliter, almost half a bottle, half a liter. But after running, it's 1,000 cubic centimeters. That is how your volume of your lungs are now expanding. The depth of breathing is increasing. Right. That's a liter, people, a liter of air that you are inhaling. Right. Also, the breaths per minute. At rest, you are having 20 breaths per minute. But look now here, after you've been running, we all know that, you know, I always want to pass out at this. If I run or walk too fast, you know, you start breathing faster, and that's what that 38 is. Okay. Explain why the amount of oxygen taken up into the blood increases after exercise. Right, let's have a look. While the rate of breathing is increased, why? Because the cellular respiration is increased. Right, remember I said you need energy, right? So the respiration rate is increased, and people, they like to combine this cellular respiration with CO2, right? So just remember that. And so there's a higher demand for oxygen and energy in the muscles. Whoopsie, what did I do now? Let me just click here again. More carbon dioxide accumulates in the blood. And when that happens, we know what happens. Rates and death of depth, not death. Yeah, well, if I run, it's death. But rate and depth of the breathing increases and your heart beats fast, and that's why you get more oxygen. Right, the second one, describe the changes that took place in the human body to bring about the breathing rate of the person. So, you know, this faster breathing over here. Right, a high concentration of CO2 led to the formation of carbonic acid. Remember I said at the beginning, when the CO2 dissolves into the bloodstream, you get carbonic acid, right? And that causes the pH of the blood to drop. Chemoreceptors in the carotid artery are stimulated by the drop in pH. Impulses are sent to the medulla oblongata. Breathing and heart rate muscles are targeted, and they will now obviously contract. Contractions will increase, and the rate and depth of breathing will also increase. Heart rate increases, more CO2 moves to the lungs to be exhaled, and therefore the blood CO2 levels will return to normal. Right. So what is important here is just to see that, just think of those steps in the mind the gap. Right. Where are they here? Right. Where are they here? That is what we are giving ticks for. But sometimes, just sometimes, the examiners ask the questions in a different way. And that's where the pH will come in, and it's not in that steps that we have in mind the gap. But sometimes they just tweak the questions a bit, and then you're like, what pH is this now? Right. But if you know the background of the story, then you will be able to answer this question. Right. And then the last question from this investigation is, 20% of the air breathed in consists of oxygen. Now, people, this is a fact that you've learned in natural science. I hope you remember this, that only 20% of the air around us is oxygen. Imagine if it was 100%. You could not smoke anything. You would explode the whole time. Even cellular respiration would be risky at that stage, right? So only 20% of our air that we breathe in consists of oxygen. Now, the air that we breathe out is 16% consisting of oxygen, right? So they want you to calculate the amount of oxygen entering the blood per breath after running. So after running, this column here. So now, this is not something we can teach you. This is now basic math. Remember, this is a science, so you need to have some common sense there and some math background, maybe up to grade 9 at least, to know that 20% minus 16% gives me 4%. So 4% was used by the body, right? And now we want to know, calculate the amount of oxygen entering the blood per breath after running. So that 4% was used by the body per breath, right? That's 1,000. Where do I get it? I get it here because that's the volume of air that after you've exercised now, running like a madman, you've got 1,000, every time you breathe in, 1,000, a liter of air. But only 4% of that is being used because it's oxygen, and that is 40 cubic centimeters or 40 milliliters. Right, so that's like a little math in there for all our math geniuses out there. Right, so that was basically the CO2 part of homeostasis. Now we are getting to the homeostatic control by the kidneys. Now, I'm going to touch on some stuff that's not necessary for grade 12, but I'm hoping that there's some grade 11 learners here as well, because I'm going to talk about some stuff that you need to know, but last year, the grade 12s, guys, you were actually doing this. So let's just test your memory banks. I know, it's rusty and, you're getting old and so on, but let's just have a look. This thing we called a nephron. Where do we get this nephron? We get it in a kidney. There are millions of them, and they're microscopic. They're very small, right? That means you can't see them with the naked eye. And they've got a very, very, very important function in our bodies, right? So, matrix, now you have to think, because I really know that you can really reach very deep and remember this, right? Firstly, it's like a little snake-like thingy. And if we look at this part here, this is the artery that goes to the nephron, and we call this the afferent arteriole, which means it's a small little blood vessel. And what comes out is the efferent arteriole. And this little arteriole goes then, can you see here? There it goes. And it's like curled around the tubules of this structure here, the nephron, right? And that's why we call it peritubular capillaries, because the periphery is around, it's around the tubes, right? So it makes it very easy for substances to diffuse out of this little tube here into the blood vessels, back into the blood vessels. And we call that reabsorption. So every time I talk about reabsorption, it's stuff going from the tube into the blood vessel. Now, what happens here, and I'm not going to go into detail about this because for grade 12, it's not important. Grade 11, it's very important for you, but I'm not going to do that today. Right, so what happens, filtration happens here. And all the stuff, the vitamins, the glucose, the waste products, everything goes into this little capsule here. We call it the Bowman capsule, right? And we call this substance then here the filtrate. And Radine was also talking about the filtrate earlier, because at the end it becomes urine, right? But a lot of things happen there. And during this part of the process, this part we call the proximal convoluted tubule, active transport happens. Now, active means it needs energy, active energy, right? So there's a lot of mitochondria here in the walls of these tubules. And what happens is at this space here, between there and there, all the glucose, the vitamins, all the useful things that the body needs. We cannot just go and urinate it out. No, you can't urinate everything out. It won't work. The body likes to recycle, right? So all those useful molecules are reabsorbed back into the blood vessel, that little peritubular capillaries, right? When we get to this little thing here, it looks like a hair clip or like a little needle thingy. That is called the loop of Henle. Now, what happens here is water starts to move out here, and that's through a process we call the sodium pump, which I will refer to a bit later on when we get to that part. I'm not going to talk about that now. And in the last part here is the distal convoluted tube, this part here. Right. And there, water is also moving out, sodium ions is also moving out. But another thing is happening here. Preservatives, maybe medicine that you drank, drugs, also moves from that blood vessel into this filtrate here. So, what happens through the whole process is, all the useful stuff is reabsorbed back into the blood. We get a situation where the water balance is now regulated. Water is also reabsorbed back into the blood, as well as water is also reabsorbed there and here as well. These are the collecting tubules, and they all come together, masses of them, and at the whole end of the story, I'm not going to go into detail here, it goes to the ureter, which goes to the bladder, and then you get urine. Okay, so that's a whole process to get urine. It's not just whoop and you drink and then it goes out. Much more going on there. Right. So what are we going to look at? We're going to look at what does this do? It regulates the water level, it regulates the pH level of the blood, and it regulates the salt level. These two, that one there, water level, and this one here, the salt level, is for grade 12. Also grade 11, but the pH level is not for grade 12. Okay, I'll just do that for interest sake for our grade 11s. Right, so this is a different way of drawing the nephron, and you can see, look at these blood vessels. They're all curled around the tubes. Right, and there you can see that's the proximal part, this is the loop of Henle part, and this is your distal convoluted tubules. Right, so the regulation of water, we call it osmoregulation. That word should sound very familiar to you. That is osmosis, the movement of water from a high water potential to a low water potential, right? And the hormone we have here is antidiuretic hormone. Now, anti means against. Diuretic means urinate. So, pee-pee. No? So, it's anti-pee-pee hormone. You don't need to go to the loo. Remember that in exams, it's going to help you. This one prevents you to go to the loo. Right. And another place where children get confused is the hypothalamus is the producer. It makes the ADH, right? But the one that secretes it is the pituitary gland. It secretes the ADH, right? So that's the dealer and that's the production or the guy who makes it, right? So let's go into regulation of water. When the when the blood has less water than normal, that is when you are dehydrated. You didn't drink enough water, you are dehydrated. Right, what happens? And remember, I get this table from the Mind the Gap. Firstly, your water in your blood decreases. What happens? Your hypothalamus picks it up. The signal is sent to your hypothalamus, and your hypothalamus will then send an impulse to the pituitary gland to secrete antidiuretic hormone. Why? You don't have enough water in your blood. You cannot afford to go to the loo now. No, you need to keep all that water in your blood. So what happens is this ADH increases the permeability of the distal convoluted tubules and the collecting tubes here. Now, permeability is a very big word. You know, I'm also English second language, and I'm like, what is permeability? It's like pores. It opens the pores so that the water can move out. So it's more permeable, so more water can be reabsorbed. Right. So what happens is it's more permeable. The water moves from the filtrate, from the tubules, into the blood vessels. And when that happens, more water is reabsorbed into your blood, and your water level in your blood returns to normal. That's what we want. Right. Now, the opposite is when you've got a lot of water in your blood. You look like a little Oros man. You're swollen. You know, you're going to explode any moment now. No. What you now need is ADH. You need to go to the loo. So ADH will help you with that. No, you don't need ADH, sorry. So the hypothalamus is stimulated once again, and impulses are sent to the pituitary gland to stop secreting that ADH. Please just stop with the ADH because you're going to explode. There's all this water in your blood, right? So this tubules here will become less permeable, right? It will allow less water to be reabsorbed back into the blood. So more water will be in this filtrate moving out to your bladder, right? So you will urinate more water out. So you will go into the loo all the time, right? To get rid of all that extra water. And then the water levels will return to normal. Right, so now the salt levels, that's a different thing. Now we have tissue fluids in our bodies, and they are affected by the presence of solid. Now solid is basically something that's dissolved in a liquid. Now our blood is liquid and our tissue fluid is liquid, and we have salts in our liquids. We need these salts for we need sodium and potassium because that regulates our nerve impulses. It helps with our muscle functioning. So if you don't have enough salt in your body, you'll have cramps, you'll have muscle cramps and your nervous system won't work so well and so on and so on. So it's very important that the salt levels are kept constant. So let's see what happens when the salt level in the blood decreases. Right, so when you don't have enough salt in your blood, your body. Firstly, the receptor cells in the afferent and efferent arterioles in the kidney will detect the low salt level. Right, so remember those little arterioles in the beginning at the Bowman capsule? They have got receptor cells in them. They are detecting now that something's wrong here. There is not enough salt. So now the adrenal glands will be stimulated. Remember where the adrenal glands are. They're on top of the kidneys, right? So they will start secreting a hormone we call aldosterone. And what aldosterone does, it increases the reabsorption of sodium ions. You cannot urinate the sodium ions out. You need it. Your body needs salt. So the sodium ions will be reabsorbed back into the blood vessels, and that will increase the salt level, and everything will return to normal, and everybody will be happy. Now, I was talking about the osmoregulation just quickly because we are running out of time. Here you can see the glucose, amino acids are all being reabsorbed over here. And what is happening here is sodium is being actively pumped out of this loop of Henry. And when that happens, this is a hypertonic solution that forms, which means this is a low water potential and this is a high water potential within this tubule. That's why water will move out passively. And this is always happening, right? But only on this side, not this side. Okay, the water will only be moving out there and over here and over here. Right. Now, what will happen if the salt level increases? Now, you've eaten too much chips, maybe a burger or so, a lot of salt in your blood, you sort of feel icky. Right, so now the receptor cells in the afferent and efferent arterioles in the kidney will detect these high salt levels. Who will be stimulated? The adrenal glands again, those little triangles on the kidneys on top, those little hats there, those glands. And they will now secrete less aldosterone. I don't like the word stop secreting, it's just secreting less aldosterone, and that will decrease the reabsorption of the sodium ions. So the sodium ions will not be reabsorbed into the blood vessel anymore. It will stay in the filtrate and move with the filtrate out to the bladder, and you will urinate it out. Right, so you will have salty urine. Right, so this is just a flow diagram like we were talking about now. If the salt levels in the blood decreases, what will happen? The adrenal gland will secrete more aldosterone, and that will reabsorb, or less sodium ions will be reabsorbed, and then the salt level will increase back to normal. And the other way around, if your salt level in the blood decreases, I'm on the wrong side, sorry. Salt level in the blood increases, adrenal glands will secrete less aldosterone, and that will, the reabsorption of sodium ions into the blood will decrease. It will rather leave with the filtrate into the bladder, and then your salt levels will decrease back to normal. It's all about getting back to normal. You don't want too much water, you don't want too much salt in your blood, too much glucose, because you can get sick and die. All right, so we don't want that. Now the pH levels I do not have time for. This is just a part, sorry, grade 11s, I wanted to touch on this, but this is not for grade 12. It's just that your pH levels must also be balanced, and this is always happening at the distal convoluted tubules where your hydrogen ions will be reabsorbed and your bicarbonate. But I'm not going to touch on that because we are running out of time. I would rather just use the last 2 minutes just to go through this question quickly. The diagram below shows reabsorption of salt and water through the tubules of a nephron in the kidney under 3 different conditions. Then they have a very nice picture here. They've got key, the black one is salt, so all the arrows here that's black is salt. Then the filtrate is the black arrow is salt, and the grey one is water. So the first one says the filtrate goes in there with salt and water. There's the filtrate going in, filtrate going in. Now they want to know, name the hormone in the human body that is responsible for controlling the water content. Oh man, people, this is so easy. Nah. We've just talked about it. You must study this. You won't have epiphany. You have to study it. ADH. Salt, aldosterone, obviously. The gland that secretes the hormone aldosterone will be the adrenal gland, And then which diagram, that's what I want to get to. Which diagram would represent a person who had eaten salty chips? Salty chips. A lot of salt in the blood. Right. On a hot day without any intake of water. So now you've got a high salt level in your blood. Very high. Right. So you need to get rid of that salt through the urine. All right. So now you cannot have a lot of reabsorption of sodium or salt. You cannot. You don't want reabsorption. You want it to stay in the filtrate. So if you look at the first one here, it's a lot of salt being reabsorbed and a lot of water. And we also don't want a lot of water to be, we want a lot of water because we didn't drink a lot of water. So a lot of water must be reabsorbed. This one, a lot of salt is being reabsorbed. Nope, we don't need that salt. Last one here, look here. A small bit, or a very small arrow indicates not a lot of salt is being reabsorbed, but a lot of water is being reabsorbed. So it's definitely diagram number 3. Okay. And with that, I have to finish off today's lesson. Unfortunately, we didn't get to everything that was prepared, but that's okay. I think you will survive. Thank you, Radine, for your part of this lesson, and thank you guys for listening. Go and study. I know you will do that tonight, and just, recap this, and see you next time.